Hi, my name is Ajay and I'm in Ms. Gentry's 5th hour biology, and I will be explaining the process of meiosis, which is a type of cell division that results in four daughter cells, each with half the number of chromosomes of the parent cell. Meiosis takes place in germ cells and consists of two parts, meiosis 1 and meiosis 2. Meiosis 1 Meiosis 1's primary purpose is to separate homologous chromosome, while meiosis 2's primary purpose is to separate sister chromatids. For the sake of simplicity here, I'll be using a cell with a diploid number of 4. The first stage of meiosis is interphase. In interphase, we have two pairs of homologous chromosomes, and the duplication of homologous chromosomes take place. In prophase 1, as you can see, we see duplicated homologous chromosomes due to the replication that occurred in interphase. Prophase 1 is characterized by three major parts. First is a disintegration of the nuclear envelope. Second is the replication of the centrosome and the formation of spindles. And third is that crossing over occurs. Paired homologs become physically connected to each other along their lengths by a zipper-like protein structure called the synaptonemal complex. When this happens, the point at which the duplicated homologous chromosomes are attached are known as the chiasmata. At the chiasmata, crossing over occurs. Crossing over is a genetic rearrangement between non-sister chromatids involving the exchange of corresponding segments of DNA molecules. Later in prophase 1, microtubules from one pole of or the other attach to the two kinetochores, protein structures at the centromeres of the two homologs. Then the homolog pairs move toward the metaphase plate. The next stage is metaphase 1. In metaphase 1, pairs of homologous chromosomes are now arranged at the metaphase plate with one chromosome in each pair facing each pole. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. Next, we have anaphase 1. In anaphase 1, the homologs move toward the opposite poles, guided by the spindle apparatus. A key characteristic of this stage is that we notice the elongation and oval-like shape of the cell. This is due to the fact that the cell is beginning to divide or move away from each other. In telophase 1, at each pole during this stage, there's a complete haploid set of chromosomes, and a cleavage furrow forms. A cleavage furrow forms because the cell is really close to dividing. This is followed by cytokinesis, which is a division of the cytoplasm which usually occurs simultaneously with telophase 1, forming two haploid daughter cells. Each haploid daughter cell consists of chromosomes that still have two sister chromatids. In some species, chromosomes decondense and nuclear envelopes form. The primary thing to know about this stage is that it is the last stage of meiosis 1 and that no chromosome duplication occurs between meiosis 1 and meiosis 2. Now we're at meiosis 2. Meiosis 2's primary purpose, as mentioned before, is to separate sister chromatids. The first stage is prophase 2. In prophase 2, as we can see, a spindle apparatus forms because of the replicated centrosomes, as you can see here. At the end of cytokinesis, each cell only used to have one centrosome, but in prophase 2, these replicate and extend their microtubules. In metaphase 2, the chromosomes are positioned at the metaphase plate like in mitosis, And because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. The next stage is anaphase 2. In anaphase 2, the spindle fibers pull chromosomes toward the pole of the cell. The next stage is telophase 2. In telophase 2, chromosomes reach opposite ends and cytokinesis occurs. In cytokinesis, the cells have completely split from each other after telophase 2, and the nuclei form, chromosomes begin condensing, chromosomes begin condensing. The meiotic division of one parent cell produces four daughter cells, each with a haploid cell of unduplicated chromosomes. The four daughter cells are genetically distinct from one another and from parent cells. And there you have it, meiosis is complete.